Welcome to the Da Vinci Academy Histology video course. The entire video course is available on YouTube and covers all of the fundamental principles of histology and relevant cell biology. You can find all of the videos from the course by clicking the histology playlist link in the description below, and then you can access the corresponding practice questions and histology lab videos by going to our website, which is also linked in the description below.
This is the fourth and the last video in the Special Sensory series and is dedicated to the macroscopic and histological overview of the ear. We'll begin with the anatomic overview of the ear. Our ears are subdivided into three segments. The external ear, which is right here that I'm circling right now, is comprised of the auricle, also known as the pinna, which is this fleshy and protruding structure on either sides of our head that are specialized to gather sounds, which channel the sound waves into the next part of the external ear, known as the external auditory meatus. This is a narrowed canal that we can stick our finger into if we so choose. The external ear ends at the tympanic membrane, this thin tissue paper-like membrane which vibrates based on the sound waves that are coming in, and it separates the external ear from the next segment of the auditory system, and that is the middle ear. This middle ear is comprised mostly of air-filled chamber and a cavity. So the middle ear begins with the tympanic membrane. It contains these three small bones called the ossicles. From lateral to medial, these ossicles are known as the malleus, incus, and stapes. And another important structure of the middle ear is the auditory tube. This auditory tube is a narrow passageway which connects the middle ear cavity to the nasopharynx. This auditory tube is usually collapsed. We can open this in order to equalize the air pressure within the middle ear cavity with the surrounding environment. So when we're taking off in an airplane or climbing up an altitude and we're feeling a little uncomfortable in our heads because of the different pressure within the middle ear, we can equalize that by opening the auditory tube, which is commonly known as popping our ears. The third and the most medial aspect of the ear is the inner ear, and this part is embedded within the petrous region of our temporal bone. The inner ear, as you can see, is really complex in its anatomy, which we'll look at in a little more detail later. But for now, just know that the inner ear has the outer bony part, and in the inside, we have really delicate membranous labyrinth that follows all the contours of the bony casing. And this membranous labyrinth is itself filled with fluid and in its entirety is kind of supported and floating within the fluid of slightly different composition. Again, more on that later. But let's first start with the histology of the external ear. The auricle, or the pinna, is covered on the external side by the skin. So we can expect to see some typical skin histology there, including hair follicles. In the inside, of course, there's dermal tissues and subdermal tissues like a lot of fatty connective tissue. But the structural support of the auricle, and the reason why our auricles maintain its shape for the most part, is due to the elastic cartilage core in the inside, which is helpful in our ability to bend our auricle and change its shape, push it against the head, but it pops right back to its original shape usually. That's thanks to the elastic cartilage. The external auditory meatus is also covered by skin, but here there are some sebaceous glands that are a little more substantial that produce cerumen, which is also known as the earwax. So the specialized sebaceous glands of the external auditory meatus are known as the ceruminous glands. In terms of the structural support that keeps the external auditory meatus open at all times is thanks to the elastic cartilage laterally. But as you can see here, towards the medial aspect, the main structural support is provided by the temporal bone. Moving on to the inner ear, we'll start with the macroscopic or anatomic overview of this complex structure. The inner ear is divided into the bony outcasing called the bony labyrinth, which is filled with fluid called perilymph. This complex anatomy of bony outcasing has several structures. These three loopy structures are called the semicircular canals because they look like semicircular tunnels. And since they're filled with fluid, we'll call them canals instead. You may note that these semicircular canals are oriented orthogonal to each other in 3 dimensions. Next, we have the vestibule, which is this bulbous area right here. The space inside the vestibule is continuous with the space within the semicircular canals, so it is still filled with perilymph. And the last part of the bony labyrinth is this snail shell-shaped structure called the cochlea. And this snail shell-looking structure makes about 2 3/4 of a turn, and the space inside it is continuous with the vestibule and is filled with the perilymph as well. You may have noticed this darker gray stuff that is inside these various regions of the bony labyrinth. These network of tubes inside the bony labyrinth is what makes the membranous labyrinth. So this long and complex tube and bulbous structures is called the membranous labyrinth, and it is filled with a different type of fluid called the endolymph. The composition of endolymph is said to be quite unique, but overall it is similar in its ionic composition to the intracellular fluid, whereas the composition of the perilymph is said to be closer to that of the interstitial fluid. Since the bony labyrinth had these 3 sub-regions, these complex tubular structures of the membranous labyrinth too have their specific regional names. The membranous labyrinth tube that runs within the semicircular canals are called the semicircular ducts. And you may have noticed that each semicircular duct ends in these ampular or slightly dilated regions, and each one of these dilated regions is called the ampulla or ampullae in plural. So we have the semicircular ducts inside the semicircular canals. Ducts in the canal is a good way to remember this arrangement. And then these bulbous ends of the semicircular ducts are called the ampullae. Within the vestibule, this large bony casing, we have 2 dilated regions of the membranous labyrinth called the utricle and saccule. Utricle is closer to the ampullae and saccule is further away from the ampullae. But still, utricle, saccule, and semicircular ducts are all continuous channels filled with endolymph. And finally, within the cochlea, we have this continuation of the membranous labyrinth in this coily tube, which is known as the cochlear duct. So cochlear duct is inside the cochlea. And within these distinct regions within the membranous labyrinth is where the specialized sensory structures are located. Within the ampulla of each semicircular duct, we have these spots which are indicating the specialized sensory structures called the cristae ampullaris. And these cristae ampullaris are specialized to sense the angular motion or position of the head. The special sensory structures of the utricle and saccule, indicated by these dark ovals, are called the maculae. The maculae within the utricle and saccule are oriented in perpendicular plane to each other, and these maculae are specialized to sense the linear position or movements of our head in either vertical or horizontal plane. And lastly, within the cochlear duct, we don't have a spot, but instead we have a really long, linear, special sensory structure that goes all the way around, following the coil of the cochlear duct. And this linear special sensory structure is called the organ of Corti. And it's this organ of Corti that is responsible for the sense of hearing. So while the cristae ampullaris and maculae of the ampullae and utricle and saccule are responsible for sense of balance and motion, it is the organ of Corti within the cochlear duct that is responsible for the sense of hearing. And it is for this reason that the inner ear is sometimes subdivided into the vestibular system for the sense of balance and the cochlear system responsible for the sense of hearing. And the cranial nerve that supplies the inner ear is also subdivided into the vestibular branch, which carries the action potential from the vestibular system, and it also has the cochlear branch, which carries the action potential from the organ of Corti of the cochlear system. Hence, the cranial nerve associated with the inner ear is named the vestibulocochlear nerve, the cranial nerve number 8. And here's the actual histology of the inner ear. And although it's not exact, I think approximately this is the plane of section through this complex three-dimensional structure. We have to play a little bit of mental gymnastics to figure this out, but based on our histological knowledge, we can see the bony labyrinth forming this bony outcasing of the inner ear. And in the inside of the bony labyrinth, we can see this network of complex tube-like structure, which is the membranous labyrinth. So this is most likely a utricle and the beginning of the semicircular duct. And this might be a semicircular duct that we cut in a cross-section. This is the perilymph-filled space outside of the membranous labyrinth, which would be filled with endolymph. So this region of the bony labyrinth would be the vestibule, and this might be the beginning of the semicircular canal. Likewise here too. And over on this side, we have the bony outcasing forming this complex rotating structure or spiraling structure. So this would be the cochlea, and in the inside, we would have this delicate network of endolymph-filled cochlear duct that has been cut in serial cross-section. So these would be the continuous cochlear duct that has been sectioned in multiple planes due to the rotating or spiraling nature throughout the cochlea. Now let's look at the specialized sensory structures of the inner ear. We'll start with the crista ampullaris. We have to remember that there are 3 ampullae, one each for the 3 semicircular ducts, and each ampulla contains the specialized sensory structure. Plural of crista ampullaris is cristae ampullaris. The crista ampullaris is comprised of the specialized hair cells with hair, which are in reality comprised of stereocilia and the specialized non-motile cilia called the kinocilium. And this structure is embedded in a gelatinous substance called the cupula. And together, the crista ampullaris is responsible for detecting the rotational position and angular movement of the head. At higher magnification of one of these crista ampullaris, we can appreciate the bony labyrinth out here. And in the inside, we have the membranous labyrinth that encloses the endolymph-filled space. On the outside of the membranous labyrinth, we would have the perilymph-filled space. And here, unfortunately, this side of the membranous labyrinth is actually collapsed inward a little bit. In reality, it should be really ballooning out. So this is an artifactual collapse. At any rate, we can appreciate that when we trace this membranous labyrinth, the lining epithelium becomes quite thick over here. And this is the crista ampullaris of the ampulla. And here, these tall cells are the hair cells, and although it's not quite as clear, you can see the apical surfaces of these cells contain these really fine hair, which are again the stereocilia and kinocilia, and these hairs are embedded in this really amorphous gelatinous substance called cupula. So imagine we are rotating our head and the endolymph moving around based on the momentum of this fluid. It would push the cupula one way or another, thereby causing the bending of these hair. And based on the amount of the bending, sometimes the action potential may fire, from which the cells may initiate an action potential, which is transferred through through some of these afferent fibers that are in touch with some of these hair cells. Next, let's look at the histology of the macula. There are two maculae in each inner ear, one in the utricle and one in the saccule. Similar to the crista ampullaris, macula is comprised of collections of hair cells with, again, hair comprised of the kinocilium and stereocilia, which are embedded in a specialized membrane called the otolithic membrane. And collectively, the maculae are responsible for detecting the linear movement of the head in either horizontal or vertical direction. We were lucky enough to catch the cross section through a utricle. So let's take a look at this area in a higher magnification, and we can appreciate this region of the membranous labyrinth that is comprised of these tall hair cells. And these hair cells, once again, have lots of these stereocilia and kinocilia that are extending out from the apical surface. And these hairs are embedded in this thin membrane that contain a lot of these heterochromatic crystalline structures that are embedded in this structure, which comprises this otolithic membrane. These crystalline or calcified structures are called the otolith, which makes this membrane slightly heavier. And once again, imagine our head moving from a stationary position and the endolymph moving in one direction or another, and based on the momentum, the otolithic membrane would move against the hair that's embedded within it, therefore bending the hair one way or another, which can then trigger an action potential which is conducted through these afferent fibers out towards the brain. And finally, the histology of the cochlea and the cochlear duct. Cochlea is a part of the bony labyrinth that is seashell shaped, and it contains in its inside this long tunnel that coils around about 2 3/4 of a turn. And in the middle of this bony cochlea, there's a bony core called the modiolus. It directly translates to the spokes, as in the spokes of a wheel. This bony core is where the spiral ganglion is located. Remember, ganglion refers to the neural tissue structure that's comprised of the neural cell bodies. And off of these neural cell bodies, we have a collection of axons that are emerging and traveling together, which forms the cochlear branch of the vestibulocochlear nerve, the cranial nerve number 8. As for the coiled tunnel, it is filled with perilymph, but in the middle of the tunnel, there is that cochlear duct. And this cochlear duct is actually anchored on either side to the bony labyrinth. So it's not a really delicate tube that is just floating without any anchorage. And due to these anchorage points, the perilymph-filled space is actually divided into 3 compartments. The scala vestibuli, that is superiorly positioned, and helicotrema is actually at the very tip of the cochlea, and the scala tympani, which is positioned below the cochlear duct. And helicotrema is where the scala vestibuli and scala tympani become continuous with each other. When I uncoil the cochlea into this long tube, what we'll learn is that the cochlear duct is positioned in the middle. And of course, the cochlear duct is filled with endolymph. And the perilymph-filled space between the cochlear duct and the bony labyrinth up here is the scala vestibuli. And below the cochlear duct is the scala tympani. And where the scala vestibuli becomes continuous with scala tympani, this little area is the helicotrema. So from the middle ear cavity, we have the oval window right here with the stapes attached. So when the sound wave comes in and is changed into the mechanical energy, the stapes kind of moves in and out against this little oval window, and this fluid will then move through the scala vestibuli, through the helicotrema, and through the scala tympani. And this force built by the pressure will be released once again into the middle ear cavity through a tiny little membranous window called a round window. And within this cochlea, we have the cochlear duct. And another name for this cochlear duct is scala media. And scala means a ladder. So it's the middle ladder, if you will. We have to remind ourselves that the cochlear duct is a part of the membranous labyrinth or membranous tube that is filled with endolymph. And it's within this cochlear duct where the organ of Corti is located, which is a specialized sensory structure that can detect sound waves and translate it into an action potential. The organ of Corti is located in this particular membrane that separates the scala media from the scala tympani. Let's look at the histology of the cochlear duct in a little more detail by zooming in onto the boxed area. This is just one section of the coiling cochlear duct within the cochlea. And at higher magnification, we can see that this is the scala vestibuli, the perilymph-filled space above the cochlear duct. And this thin membrane that separates the cochlear duct from the scala vestibuli is called the vestibular membrane. Then we have the cochlear duct, also called the scala media, hosting this specialized structure called the organ of Corti on the bottom membrane, which is known as the basilar membrane. So this basilar membrane not only has the organ of Corti, but also separates the scala media from the scala tympani. And in terms of the organ of Corti, we can appreciate that this specialized structure is comprised of these specialized sensory cells with kinocilia and stereocilia that are budding off of the inner group of cells and outer group of cells. These cells are called the inner and outer hair cells respectively. And their hair are embedded in this non-cellular membrane or structure called a tectorial membrane. So when the hair bends against the tectorial membrane, that's when the action potential is generated, which is then conducted through these neuronal fibers. And of course, the neuron cell bodies are all collected over here as a spiral ganglion. And then their distal fibers or axons are all emerging out, conducting that action potential to the brain. And the space below the basilar membrane is the scala tympani. So if I may uncoil this cochlea and cochlear duct one more time, we would see something like this again with the oval window here with stapes attached and vibrating against the scala vestibuli that's filled with perilymph. And that sound wave in the fluid would travel through, go through the helicotrema, then through the scala tympani before that pressure is released through the round window. And within the scala media or the cochlear duct, we have the organ of Corti that is all along the basilar membrane with their hair embedded in the tectorial membrane. And depending on the frequency of the sound that is being conducted through the ossicles and into the scala vestibuli and tympani, a specific region of the scala media, particularly the organ of Corti, will move just the right amount to bend the hair against the tectorial membrane, which will then trigger the action potential so that we register that sound at the accurate frequency or the pitch. What's interesting about this long coily tube is that the hair cells closer to the oval and round window happen to detect the higher frequency sound or higher pitch sound, and those that are closer to the helicotrema register more lower frequency sound. So those sounds that are a little more at the base in pitch.
Thank you for watching this video from the Da Vinci Academy Histology video course, which is completely available on YouTube. To access the corresponding practice questions and histology lab videos, go to our website using the link in the description below.